Welcome back, class. Today we start to make a transition into a new chapter and really into a new system of our body. In chapter 12, we're going to begin talking about the nervous system. Now, with the nervous system, there's obviously a couple different functions with it, but let's take a quick little overview. The nervous system really is the system of the body that controls every nerve, cell, tissue, organ of the human body. So it's really a significantly important system in regards to overall function of the human body. And so, what does the nervous system actually do? Well, the function or the main components of the nervous system is for communication and to act as a control system. The functions are very simple. It takes in information, it processes information, and then it sends initiation out to get a response. So when we look at how the nervous system collects information, we have all these receptors throughout the body, whether they detect pressure, whether they detect temperature, whether they detect sharp or soft stimuli. There's all different types of sensations that these particular types of cells and receptors detect. And so what they do is they detect these stimuli, and then they send that information through the nerves into the spinal cord and up to the brain. When it gets to the brain, and while it's in the spinal cord at the same time, it actually processes this information. And then when it gets to the brain, it evaluates it and interprets it. And it helps to determine what type of response to act on as a result of that sensory input. We then have to take that information and send it back out to the rest of the body. And so we then initiate a response to the information that we received and processed. And this sends information out to the effectors. And our effectors can be our muscles or our glands, or really any other tissue that actually is going to perform a particular response. So when we look at the organization of the nervous system, we really are able to break it down. And we start at the very top as of the nervous system as a whole. And then when we look at the structural organization, we break it down into the central nervous system and the peripheral nervous system. The central nervous system is pretty simple. It's the brain and the spinal cord. So we have the brain in your skull, the spinal cord going down your vertebral column. Now, the peripheral nervous system, or the PNS, involves the nerves and the ganglia. So anything that comes off of the spinal cord, and I guess including the brain when you get to cranial nerves, those are all part of the peripheral nervous system. And we'll talk about what ganglia are here in just a few minutes. Now, as we look at the organization of the nervous system, again, we look at the nervous system as a whole, and we really start at the top. We start with the central nervous system essentially going down the left side. We look at the peripheral nervous system going down the right side. And so we essentially are breaking down the right side of the nervous system. So I am going to kind of draw this tree here up at the top. And this is what I mean by that. We look at the nervous system as being at the very top. We go down to the central nervous system and the peripheral nervous system. And when we do that, we now are able to separate the peripheral nervous system into the following: the sensory nervous system and the motor nervous system. And we'll subdivide the motor nervous system here in just a second. But let's talk about these functional components here. Let's look at sensory versus motor. When we look at the sensory nervous system, this is what we call the afferent nervous system. So again, we've talked about this before. Afferent refers to sensory. This is the part of the nervous system that receives information from the receptors and transmits it towards the CNS, which means we are in the PNS during this point. We now move to the CNS, we get into the spinal cord, and this takes the information up to the brain before we hit the motor nervous system and sending information back out. When we look at the sensory nervous system, we can break this down into more specific components. We have the somatic sensory nervous system. And if you remember, there's a couple things that we're supposed to think of when we think of or hear somatic. It should be skin, skeletal muscle, and joint. So when we look at the skin, skeletal muscle, and joint component, the somatic sensory system, it's going to detect stimuli that we consciously perceive. So we consciously perceive motion. We consciously perceive feelings on our skin. We consciously perceive the stretch that we get in muscle tissue. So again, the sense or the somatic system is where we are detecting stimuli that we consciously perceive. On the other hand, the visceral sensory system detects stimuli that we typically do not consciously perceive. So you may want to put the word consciously in there. But this is where we don't consciously feel it. So things like the heart or the kidneys. Yeah, we may detect some discomfort and pain in certain instances, but as far as the visceral component, we don't feel what's going on in the kidneys, but yet they still function. We don't necessarily feel all the things going on in the heart, but it still functions. So this is the visceral sensory system. We don't have to actually know what's going on for it to continue to work. Now, if we go down the other side of this chart and we get into the motor nervous system, we're going down this side. This is the efferent system. Again, we've talked about this before as well. Efferent refers to motor. So with the motor nervous system, this initiates the motor output and it actually transmits it from the CNS towards the effectors. So the motor component, the motor signal starts in the brain, goes down the brain, down the spinal cord, and then out into the rest of the body in order to initiate some sort of response. Again, we have this division of the motor nervous system, just as we did with the sensory nervous system. And we have the somatic motor system, and it's going to send voluntary signals to the skeletal muscle to cause voluntary movements. The autonomic motor system is the visceral motor system, and it sends involuntary commands to the heart, smooth muscle, and glands, basically in an attempt to control your visceral organs. And it does have parasympathetic and sympathetic divisions. So again, we get up into this chart here, and we follow this from PNS to sensory system and motor system. And then our motor nervous system is then divided into sympathetic and parasympathetic divisions. And that's going to give us different responses in the motor pathway. So here we kind of have this division or this flow chart, if you will. These just show us our functional divisions of the nervous system for the body. We have our sensory nervous system or the input system. And then we have the output or the motor nervous system. With the input system, we have the somatic sensory and the visceral sensory. With the output system, we have the somatic motor and the autonomic motor. Okay? And again, we will have this autonomic motor system subdivided into sympathetic and parasympathetic divisions. But you can go ahead and just take a look at this, review it, kind of understand the divisions and the functional organization here. So now we get into different components of the nerves and the things that are involved with the nervous system. And so one of the first things we're looking at are things that are involved in the PNS. So these are things in the peripheral nervous system. So the first thing we're going to look at is a nerve. Now, I will tell you, nerves and ganglia, these are going to be two terms that you want to know because I am going to use those quite a bit as we get into later chapters. And so you just want to have a good understanding of what they are. Plus, they will also be on your quizzes as well as your exams and final exam. So when we look at a nerve is simply put a bundle of parallel axons in the PNS. Now understand that the axon is an extension coming off of the cell body of a nerve cell. And so when we take a bunch of these axons and a bunch of these processes, they become a bundle. And as we put them all together, they form a nerve. And so these are a bundle of parallel axons found within the PNS. So I'll just go ahead and circle or highlight that and circle it for you. We also have to look at the connective tissue wrappings. So just as in the muscle tissue, it had connective tissue coverings, we have the same thing with the nerves. And you'll notice that the prefix on all of these is essentially the same as what it was with the muscle tissue. And the organization itself is very, very similar to what it was with muscle tissue. So we have an epineurium, a perineurium, and an endoneurium. An epineurium encloses the entire nerve, just as the epimysium, encloses the entire muscle. The epineurium is a thick layer of dense, irregular connective tissue. The perineurium wraps the fascicle, which again is very similar to what we have with the paramecium. The perineurium wraps the fascicle, which is a bundle of axons within a nerve. And so think of the axons as part of the cell. So it's basically wrapping individual collections of these cells together the same way that we see with muscle tissue. And the perineurium is a layer of dense, irregular connective tissue. Again, all of these are connective tissue until we get to the endoneurium, which is still connective tissue, but now it's a very delicate layer of connective tissue. And the endoneurium wraps an individual axon. Again, just as the endomysium wraps the individual muscle cell. And this helps to separate and electrically insulate each axon, just as we did with the muscle cell. Now, one thing that we know, our nerves are vascularized. So we do have blood vessels going to them. And those blood vessels are able to branch through the epineurium and the perineurium to become capillaries. Again, we all know capillaries are these very small vessels that are able to provide a width of about 1 or 2 red blood cells. But this does allow for the exchange of gases between axons and the bloodstream. So now we get movement of oxygen and ions all within this area. So if we look at the structure of the nerve here, again, we have all our connective tissue. We have the epineurium that covers the outside. Here we have a fascicle, just as well as we have here. And this is the perineurium that covers the fascicle. If we pull out one of these individual structures, this is the axon. And you'll notice, I know it's going to be very difficult to see for you, but there are little bubbles of white. Those indicate myelination, and we'll talk about that coming up as well. But then we see our endoneurium, which again is this very delicate connective tissue that surrounds the cell and the axon itself. If we look down here, we'll talk more about this when we get into the spinal cord, but you'll see the actual structure here of the cord itself. This butterfly-looking structure is going to be called our gray matter core. And we'll spend plenty of time on that when we get into the CNS chapter. So if we look at some of the structural classifications of nerves, we have the cranial nerve or cranial nerves, and these are actually originating in the brain, with the exception of cranial nerve 11, and we'll talk about that one. Spinal nerves, on the other hand, extend from the spinal cord. And so these are all, again, part of the PNS. Now, what is the function of nerves? Well, or the functional classifications of nerves? Well, basically, we look at the function of what that nerve does. Does it take in information, or does it send out information? Or does it do both? So a sensory nerve contains sensory neurons sending signals towards the CNS. So sensory nerves are afferent fibers, or collections of afferent fibers. Motor nerves, on the other hand, contain motor neurons. These contain efferent fibers, and they send signals from the CNS out to the rest of the body. We can also have what we call a mixed nerve. And as you guessed, a mixed nerve contains both sensory and motor neurons. Most named nerves in our entire body are mixed, which means they have dual function. Most of them are going to take in information to the brain, but then they're also going to send information back out to the rest of the body. We do have some individual neurons, or some individual nerves, I should say, that are going to be only motor or only sensory, but those are not the majority. Individual axons in these nerves, however, transmit only one type of information. So we can have 1,000 individual axons, but of those 1,000 axons, there's only going to be so many that take information in and so many that send information out. It's not a dual function, okay? We are not sending information along one neuron out to the body and then information coming back in on that same pathway. We only have information going in one direction on individual neurons and individual axons. Now, the other component of the PNS is a ganglion. And a ganglion simply is a cluster of neuron cell bodies that we find within the PNS. So again, this is another one of those definitions, a bolded term, that we do want to know what the definition is. So again, it's a cluster of neuron cell bodies that we find in the PNS. So let's look at a few characteristics of a neuron. Well, one, a neuron has to be excitable, which means it has to respond to a stimulus. We have a particular stimuli that comes in, and just as we have with muscle tissue, how it changes its voltage in response to the signal, this is what happens with a neuron as well. The stimulus itself is going to cause a change in the cell membrane's potential, and we're going to see a flood of ions causing the cell to change its polarity or change its voltage. It also has to have conductivity, which means we have to conduct a particular electrical signal. We have to be able to propagate and send that electrical signal down the cell. And it does this through voltage-gated channels along the membrane. And so these channels open sequentially, again, just as we do with muscle tissue. So you're kind of starting to see a trend that the muscle cell is going to have a lot of very similar characteristics to what we see with our neurons. It's got to have a secretory component, which means it has to be able to release the neurotransmitter in response to some sort of conductive activity. We see this at the neuromuscular junction with the axon terminal. That is a part of the neuron, and so it releases the neurotransmitter into the synaptic cleft. And so this neurotransmitter is the messenger that gets released from the vesicle, and it influences the target cell. Maybe it causes some sort of excitability, or maybe it causes some sort of inhibitory component to the target cell. Our next characteristic of neurons is that they display extreme longevity, meaning they basically can live throughout a person's lifetime. Now, a term that kind of gives that extreme longevity is called amitotic. So our neurons, because of their amitotic characteristic, we will notice that after fetal development, we really lose mitotic activity. And so because mitotic activity is lost in the neurons, amitotic basically means that neurons can't be replaced. So at this point, when a cell or when a neuron itself dies, it basically means that it's gone forever. So amitotic means a cell cannot be replaced. So let's start taking a look at the neuron itself. When we look at a neuron, we have several significant parts of that neuron. The first one is the cell body. Now, another name for a cell body is going to be called a soma. Now, just with all other cells, we have a plasma membrane in this particular instance, and it encloses the cytoplasm just like most all other cells. A neuron does contain a nucleus, and it's the cell body really that initiates some of our graded potentials, which we'll talk about, but it is also going to receive other potentials from our dendrites. And it helps to conduct these potentials down to the axon. Our dendrites are going to be the short, unmyelinated processes that branch off the cell body. This is the receptive area of the cell, and it receives input and transfers it to the cell body. Now you'll notice this term up here, unmyelinated. So when we talk about myelinated versus unmyelinated, myelinated is basically the insulation, and it allows the cell or the neuron to conduct the signal at a faster rate. Unmyelinated means that there is no myelination at this location, and it causes the transfer of the signal down the cell to be a little bit slower. And we will talk about that a little bit later, but just to kind of give you an idea of what these terms mean already. Then we get to the axon. And you'll notice that this basically takes up an entire slide because the axon is probably the most significant part of the neuron structure, especially when it comes to function. So the axon is the long process that emanates from the cell body. It makes contact with other neurons, muscle cells, or glands. And it does this by causing or creating what we know as a synapse. It's going to attach to the cell body at an axon hillock. And this axon hillock is kind of this triangular region of the cell body. The cytoplasm within the axon itself is going to be called the axoplasm, and the membrane is going to be called the axolemma. So we have the cytoplasm known as the axoplasm, and the plasma membrane of the axon is going to be called the axolemma. As we go down the axon, we're going to have little branches coming off of it, and these are called axon collaterals. As we get to the very end of the axon, we get to the axon terminal, and it can cause the creation of several axon terminals, and these are called telodendria. Now, when we get to the tips or the ends of these axon terminals, these are what we call synaptic nodes. And if you remember going back to the neuromuscular junction, we had the axon coming down, we had the axon terminal, and we had this kind of like bubble shape. That bubble shape basically is what we know as the synaptic knob. And within the synaptic knob and within these axon terminals, we're going to see these synaptic vesicles. And the synaptic vesicles are the structures that are going to contain our neurotransmitters. Now, this could be acetylcholine, it could be norepinephrine, or it could be a collection of numerous other different types of neurotransmitters. The axon's function is to conduct action potentials and then release the neurotransmitter at the synaptic knob. So this is the output region of the neuron. So just as the dendrites were the input region coming in toward the cell body, the axon takes information away from the cell body. So again, this becomes the output region. Our next part of the neuron is the cytoskeleton. Now, the cytoskeleton is going to be composed of our microfilaments, intermediate filaments, and microtubules. And these basically help to provide the skeleton of the cell, essentially giving it structure within the cytoplasm. The intermediate filaments are going to be termed neurofilaments, and they aggregate to form bundles called neurofibrils. And these neurofibrils basically are providing the tensile strength of the neuron itself. So let's take a look here at the neuron. And so you see we have it drawn in graphical representation, but then we also have a nice microscopic view of it as well. So here we first have the nucleus found in the middle of the cell body. This whole structure here is going to be our cell body. And this includes going all the way down here into the axon hillock. These are going to be our dendrites. This is the input region. You'll notice here we have a axon that's coming down and connecting to this other neuron. These are forming synapses at each location. Here you'll notice that we have this red box that we blew up and pulled out. This is the synapse, or a connection between the neuron and another neuron. If we go down into the axon hillock, here is your triangular region. So we have this triangular region that develops as it leads from the cell body into the axon. As we move down, we have the axon component of it. Here is a split off called an axon collateral. And if we go all the way down here, you'll notice that we have all these split offs. These are going to be our terminal extensions, or what we're getting down to our telodendria. If we also look at the blow up component of it, you'll also see here's our axon terminal with our synaptic knob, with our synaptic vesicles inside. And those synaptic vesicles are containing our neurotransmitters. So again, this is a really good graphic to at least look at, to learn and understand the structure of the neuron itself. And if it helps, remember, looking at the connective tissue coverings, we would essentially have numerous axons coming down through here that are coming from numerous neurons. And that's where our perineurium, epineurium, endoneurium all surround. This is going to be surrounded by our endoneurium. If we take a bunch of these axons together and form a fascicle, that's going to be surrounded by our perineurium. And then if we take that and create a nerve, then we have our epineurium. So now we start looking at how neurons transport structures or substances within the cell itself. So we have what we call anterograde transport and retrograde transport. Anterograde transport is from the cell body. So basically we're moving things from the cell body down the axon. And so it moves newly synthesized material toward our synaptic knobs. If we look at retrograde transport, this is going toward the cell body. So we're basically looking at materials from the axon that were broken down or used in recycling. And this is moving from the axon toward the cell body. And this can occur by fast axonal transport or slow axonal transport. Obviously, fast is going to be at a little higher rate of speed as compared to slow. So if we look at fast axonal transport, it's going to occur at about 400 millimeters per day. So understand, it's still not very fast. So an entire day we can move things 400 millimeters. It involves the movement along the microtubules. And it's going to be powered by motor proteins that utilize and split ATP. Understand that with fast axonal transport, we can have things moving in an anterograde or a retrograde motion. So we can have things moving toward the cell body or away from the cell body. If we look at anterograde transport, we're really looking at things like vesicles, organelles, and glycoproteins. And when we look at retrograde transport, we're really looking at used vesicles, potentially harmful agents, things that basically need broken down. So retrograde is more stuff that's bad and negative. Anterograde is things that the body and cell need. Slow axonal transport occurs at about 0.1 to 3 millimeters per day. So obviously significantly slower. And it's going to result from the flow of the axoplasm. Now, substances only move from the cell body toward the synaptic knob are going to be things that include enzymes, cytoskeletal components, and new axoplasm. So now how do we classify neurons based on their structure? So we classified neurons based on their function, whether they were sensory or motor. But now we also look at the structural classification, and we do this by the number of processes that come off of the cell body. So we have a multipolar neuron. With a multipolar neuron, we have many, many dendrites that come into the cell body, but we only have one axon coming off of the cell body. This is the most common type of neuron in the human body. It makes up approximately 99% or just over 99% of all of the neurons that we have. Bipolar neurons are going to have one dendrite and one axon. So essentially we have two processes, but one that comes in and one that goes out. These are relatively uncommon, and we find them in the retina of the eye. Now, unipolar neurons, or what we call pseudo-unipolar, basically has one process that extends from the cell body. However, that one process will actually split off into two separate processes. Now, one of those processes is called the peripheral process, and it's going to split into several receptive dendrites. So again, this is the part that goes out into the periphery of the body. Now, most of these are going to be sensory neurons. And so the peripheral process essentially is more the receptive end of this particular part of the cell. The central process is the other one, and it leads to the synaptic knobs in the CNS. So it's going from the cell body then into more of the CNS area. And so the information starts at the peripheral process, comes into the cell body, and then goes back out into the central process towards the CNS. Now, when we look at the functional classification, we really talked about functional classifications a little bit already. But their classification is based on the direction they propagate their action potentials. So our first functional classification is the sensory neuron. These are afferent neurons. They conduct input from the somatic and visceral receptors towards the CNS. Most of these, again, are unipolar, okay? So they relate to our structural classification, although a few of them are bipolar. Then we have our motor neurons. These are the efferent neurons, and they conduct input from the CNS out to the somatic and visceral effectors. All of these types of neurons are multipolar. Well, then we have a third type of functional neuron, and it's called an interneuron, and these are what we know as association neurons. They're going to receive, process, and integrate information from many other neurons throughout the body. These neurons are responsible for communicating between the sensory and the motor neurons. Interneurons are located within the CNS, which means they are in the brain or in the spinal cord, and they make up about 99% of our functional classification of neurons, and these are generally multipolar. So if we look at motor neurons being multipolar and interneurons being multipolar, it coincides with the fact that interneurons make up 99% and multipolar neurons also make up about 99% of our neurons. So if we look at the functional component here we have our receptors. They come in this peripheral process of our unipolar cell. Here's the cell body of our unipolar cell. And then here's our central process as it leads into the central nervous system. Here's our interneuron. It allows us to communicate with sensory and motor information. And so it bridges the gap. It allows us to create a response. And then here we have our motor neuron, which again is a type of multipolar. Here's our axon as it goes out the spinal cord and out to the rest of the body. And in this case, synapsing with a skeletal muscle. So now let's look at the synapses and how neurons connect with each other. So a synapse is simply a place where a neuron connects to another neuron or an effector. Now there are two different types of synapses. We have a chemical synapse and an electrical synapse. A chemical synapse are far more common than electrical synapses. So chemical synapses are the ones that we've already talked about. It's what we see at the neuromuscular junction where there's a gap and we use a chemical. If we look at an electrical synapse, we have presynaptic and postsynaptic neurons, and those are bound together by gap junctions. The thing about an electrical synapse is the pathway is very quick because there is no synaptic delay in the response of the neurotransmitter having to cross the synaptic cleft. And so there's no delay in passing of the electrical signal because it goes directly from one cell to the next through those gap junctions. Now, the chemical synapse, on the other hand, is the one that, again, we have talked about quite a bit. We have the presynaptic neuron, and that presynaptic neuron axon terminal produces the signal by releasing the neurotransmitter. So we have the action potential coming down the presynaptic neuron, reaches the axon terminal, where it causes the release of the neurotransmitter. The postsynaptic neuron actually receives the signal from the presynaptic neuron. The postsynaptic neuron is most commonly with one of the dendrites on the postsynaptic cell. So you'll notice that we have some terminology here with presynaptic and postsynaptic. When we talk about presynaptic, we're talking about before the synapse. Postsynaptic refers to after the synapse. The synaptic cleft, which we've already talked about, is the small fluid-filled gap that occurs between the two neurons. And because we have a space, this is not this is not a direct connection. It's an indirect connection because the two cells are not physically in contact with each other. But if we do look at the events of this synaptic communication, again, it's really the things that we've already talked about with muscle contraction. The neurotransmitter molecules are released from the vesicles of the synaptic knob into the cleft as a response to an action potential that arrived on the presynaptic neuron and at the presynaptic axon terminal. The neurotransmitter then diffuses across the cleft and binds to the postsynaptic receptors, which are going to be called ligand-gated receptors. The binding of this neurotransmitter to the receptor then initiates a postsynaptic potential, which we know as a graded potential or a type of graded potential. Now understand that we'll talk about the difference here, but I'm going to give you the difference. An action potential is an all or none event. Once we reach threshold, it's going to spike all the way to positive 30 as far as positive 30 millivolts. With a graded potential, it's a little bit smaller and it may not reach threshold. And if it doesn't reach threshold, it's not enough or it's not strong enough to generate an action potential. Now, the crossing of the neurotransmitter across the synaptic cleft, these components are what we know as synaptic delay. Because synaptic delay is the time that it takes the neurotransmitter to be released, the neurotransmitter to cross the cleft, and the neurotransmitter to bind to the receptor. These three phases or these three steps are all a part of what we know as synaptic delay. So now that we've talked about the neuron, now let's talk about some other nerve cells that we find within the nervous system. Our first type or our major type is going to be what we know as glial cells. Now, glial cells are non-excitable cells, so they don't actually pass an action potential like our nerve cells or like our neurons do. But they do support the cells found in the CNS and the PNS. Now, they are smaller than our neurons, but they far outnumber them, and they account for about half of the volume of the entire nervous system. These cells are capable of mitosis, and they help to protect and nourish the neurons themselves so that the neurons are able to continuously function the way they're supposed to. They basically are providing the physical scaffolding for nervous tissue. And so these are providing support and allowing the nerve cells or the neurons to perform their functions, but they also help to guide and migrate the neurons during development to essentially put them in the position that they are designed for. And our glial cells are critical for normal function at the neural synapses. So our first type of glial cell is an astrocyte. Now, an astrocyte is a star-shaped cell. It's going to have our processes that basically end in what we know as, or what we call, perivascular feet. These are the most abundant glial cells in the CNS. And because of their structure, they actually help to form the blood-brain barrier. And it uses these perivascular feet by wrapping around the capillaries that we find in and around our brain. And by wrapping and forming this barrier, it actually controls the amount of substances or even the type of substances that have access to our brain tissue. Astrocytes are also responsible for helping to regulate tissue fluid composition, and it can help to regulate the potassium concentration that we find in this chemical environment around our neurons. They're also there for structural support. So just as some of these neurons will wrap around the capillaries to form the blood-brain barrier, they're also going to extend out and essentially wrap around the neurons in order to form this structural support. They do assist in neuronal development, and then they also help to occupy the space of dying neurons. Because remember, our neurons are amitotic, so the neurons themselves cannot be replaced if the cell body has been damaged. Our second type of cell for glial cells is an ependymal cell. Now, ependymal cells line the cavities in the brain and the spinal cord, and they help to form part of the choroid plexus, which helps to produce the cerebrospinal fluid, or CSF. We're also going to notice that ependymal cells are oftentimes, or what we see, they're ciliated. And so these are going to have cilia that help to move CSF within the cavities as well. Now, microglia are small cells that wander the CNS and help to replicate in an infection. So these are basically the immune system cells. They act as phagocytic cells within the nerve system, or particularly within the CNS. These are going to engulf our infectious agents and help to remove debris. Our fourth type of glial cell is an oligodendrocyte. Now, oligodendrocytes are large cells with slender extensions. And as they extend out, they're going to wrap around the axons of the neurons, and they help to form the myelin sheath. Now, these first four types of cells, the glial cells that we've talked about, are astrocytes, as I scroll back here. Our ependymal cells, our microglia, and our oligodendrocytes are all a part of the CNS, okay? So these are glial cells found within the CNS. We'll talk about two separate types of cells that we find in the PNS coming up. But this will be a good spot to add our attendance code word. And let's just make that glial cells. So our attendance code words for today are going to be glial cells. Now, this image will be a really good image for you to look at and review as far as what the cells are, what some of their functions are, because again, it does have them listed here. But again, know that these are the cells, or these are the glial cells that we find within the CNS. And so please take some time to hit pause here, go ahead and review what you need to, and then you're able to continue on. So now we look at some of the glial cells of the PNS. We have to look at our satellite cells first. And our satellite cells are arranged around our neuronal cell bodies within a ganglion. Remember, ganglion are collections of cell bodies found within the PNS. They electrically insulate and regulate the exchange of nutrients and waste, again, in and around the cell body. Our second type of glial cell is a neurolemmacyte, or what we call a Schwann cell. Our Schwann cells are elongated flat cells that ensheath or enclose our PNS axons with myelin. And by myelinating these cells, it allows for faster action potential propagation and allows us to send that signal down those axons and from cell to cell at a higher rate of speed. So we take a look at the glial cells of our peripheral nervous system. Here we have our neuron. This is a unipolar cell. Here we have our satellite cell. And it, again, surrounds the cell body of our neuron. Now here, we look down at the process. In this case, this is a motor neurons. So we have the output component or an efferent pathway. And you'll see that we have the neurolemmacytes that basically form these concentric rings. And it forms this myelin sheath, of which we have a little bit of a space at each little area between each cell. We'll talk about those spaces, which are known as nodes of Ranvier, and we'll talk about what the benefits of having those are and how they actually increase the rate of speed. But you'll see that we do have these Schwann cells also on this sensory neuron. So let's look at a clinical view of our nervous system up to this point. And we start to look at tumors. Now, tumors essentially are neoplasms from some unregulated cell growth. And these are what we refer to as tumors. They sometimes occur in the CNS, and tumors originating from the brain are what we know as primary brain tumors. Typically, they're going to originate in our supporting tissues, and these are going to include a lot of our glial cells. And the reason this occurs is because our glial cells are the cells that have the capability and the capacity to undergo mitosis. And so tumors essentially exploit that ability and cause some of those cells to speed up that mitosis or that mitotic activity. We also see that they can develop from the meninges. And these are the tissues that form a protective membrane of the CNS, or they can occur in our glial cells. Glial cell tumors are what we know as gliomas. They can be relatively benign, which means we can just pull them out and hopefully have no issues with them. Or they can be malignant, which means they're cancerous, and they're capable of metastasizing and spreading throughout the rest of the body. So let's talk a little bit about myelination. So we've kind of talked about the two types of cells that do myelination. If we're talking about myelination in the CNS, it's our oligodendrocytes. If we're talking about myelination in our PNS, these are the Schwann cells or the neurolemmacytes. So the myelin is essentially several layers of the membrane of glial cells. It's this wrapping of the axon with the myelin cells. Now, myelin has a very high lipid content, and it gives the neuron or the axon kind of a glossy white appearance, and it insulates the axon. The glia are the neurolemmacytes in the PNS, as we said, and there are the oligodendrocytes in the CNS. Now, if we look at it in the PNS, the neurolemmacytes encircle the neuron axon, and it wraps it in these concentric layers, forming the myelin sheath. The neurolemmacyte cytoplasm and nucleus are actually then pushed to the periphery, forming what we know as a neurolemma. Now, a neurolemmacyte or a Schwann cell can myelinate only about 1 millimeter of an axon. So in order to myelinate the entire axon, it's going to take several Schwann cells. And then because of that, because we're utilizing different cells along the entire axon, we form these gaps. And these gaps, again, as I just said, are called nodes of Ranvier. So if we look at the myelination of our PNS axons, we see this ring starting to form around. And you'll see that it just continues to wrap and wrap and wrap as it goes all the way around. And again, just forms this concentric ring as the inside of the layers begin to flatten out. And as it forms it, or as it wraps around, this is what we then know as a part of the myelin sheath. And again, it's going to do this throughout the entire length of a particular axon. Take a look at the PNS and we take a little bit bigger look at it. You'll see that each of these is a large, or not large, but is a neurolemmacyte or a Schwann cell. And then the space in between there, again, is the node of Ranvier. And again, we'll talk about how that actually speeds up the process of the action potential. So if we look at the oligodendrocyte then in the CNS, this is an image of our oligodendrocyte, and you'll see the feet here as it extends out from the cell body into or onto the neuron or the axons of these particular neurons. And it'll do the same thing. The extension extends out and it's going to continue to wrap around, forming this myelin sheath, also creating, again, these nodes of Ranvier, even in the CNS. You'll note that one oligodendrocyte can myelinate 1 millimeter of multiple axons. No neurolemma is formed because, again, those will be found more so in the PNS. And the nodes of Ranvier are still present between the adjacent wrapped segments. When we look at unmyelinated axons in the PNS, you'll notice that the axons sits in depressed portions of the neurolemma site. And so the neurolemma site doesn't actually completely go all the way around, and then the individual axons themselves aren't myelinated as well. And so you'll see that the neurolemma site starts to envelope multiple axons, but in essence isn't completely surrounded. In the CNS, unmyelin axons are not associated with the oligodendrocytes. And so the unmyelinated axons are enveloped by the neurolemma site, but there are no myelin sheath layers around each individual axon. So we don't always have to have myelinated cells in the PNS and the CNS. And in some cases, these cells are actually going to be unmyelinated. So let's look at a clinical view of the nervous system of some disorders that actually affect the myelin of the neuron. And that condition really is multiple sclerosis. MS is kind of a progressive demyelination condition of the neurons in the CNS. It's really an autoimmune disorder, and it's where the oligodendrocytes are attacked by immune cells. This attack causes kind of a repeated inflammatory event, and what it does then is it causes scarring and permanent loss of function of these neurons. And so we see this kind of progressive effect as we continue in the condition itself. Now, axons have the ability to regenerate themselves. Now, we talked about how a neuron itself is amitotic and cannot be replaced, but that refers to the cell body of the neuron. If the cell body is damaged, it basically means that the neuron is essentially dead. But that doesn't mean that we can't, in a sense, regenerate axons in neurons where the cell bodies are still intact. So after certain traumatic injuries, the PNS axons can regenerate themselves. And you'll see here that regeneration is possible if the neuron cell body is still intact and there's enough of the neurolemma that remains. Now, regeneration success is more likely if the amount of damage is less extensive and the distance between the site of damage and the structure it innervates is a relatively shorter distance. Obviously, the longer the distance or the greater the distance, the more difficult it is for the cells or the axons to regenerate themselves. So let's look at some of these steps here that are involved in axon regeneration. In the first instance here, we have the axon gets severed by some sort of trauma. Now, if we look at proximal to that injury, the axon is going to seal off and swell. If we look distal to the cut, this is where the axon and the sheath are going to degenerate. And this is called Wallerian degeneration. But we're also going to see that the neurolemma itself still survives. The neurolemma then and the endoneurium form a regeneration tube. And this allows the axon to regenerate guided by certain nerve growth factors that are released by the neurolemmacytes. And the axon then is going to reinnervate its original effector or its initial or original sensory receptor. So here's kind of these steps. You can take some time and go through them. It's relatively small here. So take some time and look this up in your book if you want to, or if you feel like you need to. And you'll be able to see that there's the injury. You'll see what happens proximal to the cut versus distal to the cut. You'll see that the regeneration tube is occurring or developing. You'll see that the neuron is able to regenerate. And here we have this connection then back to the effector of the cell. So again, please make sure you spend some time and just kind of review that. Now, the CNS axon regeneration is actually limited. So what we saw before in the previous two slides was really involving more PNS regeneration. Because when we look at the CNS, the oligodendrocytes actually secrete growth inhibiting molecules. And so what we see is where large numbers of axons are going to crowd into the CNS. And so regrowth is obstructed by some of the scars from the astrocytes and the connective tissue. Remember, we said astrocytes kind of fill in the areas where scarring has taken place or where tissue has been damaged. So as we continue to look at the neurons, we now start to look at how some of the neurons are going to start to utilize pumps and channels within their tissue. We're going to find that these pumps or these membrane proteins help to maintain a concentration gradient by moving substances against that particular gradient. Remember, these are utilizing energy if they are moving against their gradient. Because of this, it requires cellular energy, and the neurons themselves are going to have sodium-potassium pumps as well as calcium pumps in their membranes. Because again, we're going to have to rebalance our sodium and potassium after we've gone through an action potential. But we also are going to have to remove or repump different calcium ions, again, trying to help balance out its concentration gradients as well. Now, if we look at channels then, channels are these protein pores within the membrane that allow ions to move down their concentration gradients. This allows that to move into or out of the cell. And when they open, they allow only a specific type of ion to diffuse. If we have a leak or a leakage channel, these are a type of passive channels, and they're always open for continuous diffusion. With chemically gated channels, these are normally closed. However, they open when a transmitter binds to it. And these are the channels that we see within the synapse, as we see in the case of the neuromuscular junction, where we found that acetylcholine crossed the synaptic cleft and bound to our ligand-gated sodium-potassium channel. Voltage channels are also normally closed, but they do open when the membrane charge changes. These are what we see with sodium voltage-gated channels, potassium voltage-gated channels, and calcium voltage-gated channels. They all open in response to a particular voltage. Now, there are three states of a voltage-gated sodium channel. These are the channels that help to propagate the action potential as we leave the synaptic cleft on the neuron or on the muscle cell. We have the resting state. Now, we'll notice that with the sodium channels, the activation gate is closed and the inactivation gate is open. So with a sodium voltage-gated channel, there's going to be two particular gates that help to block this off. Again, the activation gate is closed at resting, and the inactivation gate is open. And it's at this point that the entry of sodium is prevented. During the activation state, the activation gates open, and this is due to the voltage change. The inactivation gate remains open as well. And so at this point then, sodium is able to move through the channel. Now, during the inactivation state, the activation gate still remains open, but we see the inactivation gate begins to close. And because we at least have one closed gate, the entry of sodium is also prevented in this situation. This state tends to last for a very short time, and then once this process ends, the channel quickly resets to its resting state, which therefore then means the inactivation gate begins to open and the activation gate closes, and so we're still in a closed state. So when we're in the resting state and the inactivation state, that prevents the entry of sodium. It's only in the activation state do we have the ability to allow sodium to move into the channel or into the cell. So because of the voltage gated sodium channels, two gates, there actually can be three states of which we just talked about. And so you'll see here we have the activation gate closed, in this case the inactivation gate open. In this particular state, in the activation phase, the activation gate is open. The inactivation gate is also open, allowing sodium to move into the cell. And then we get into the inactivation phase. This is where the inactivation gate is closed, the activation gate is open, but that also closes off the channel, meaning no sodium can get in. If we look at the plasma membrane of a neuron, we have a very wide distribution of pumps and channels. We know we have a bunch of leakage channels. We have sodium leakage channels. We have potassium leakage channels. We also have our sodium potassium pumps. These collections, or the collections of all of these, the leakage channels, the sodium potassium pumps, all help to maintain the resting membrane potential of that minus 70 millivolts across a muscle cell, or that same voltage across a neuron. When we look at the plasma membrane, we have a whole bunch of functional segments within the neuron. And we kind of touched on these a little bit so far. We have the receptive region, which is the dendrite in the cell body. And these involve chemically gated channels, which include chemically gated chlorine channels, which we really haven't talked about much at all. We have the initial region, which is the axon hillock. And this initial region is the area of the cell that actually initiates the action potential after all of our graded potentials have arrived around our dendrites in our cell body. It's at the initial region that we have our voltage-gated sodium channels and our voltage-gated potassium channels. The conductive region is the axon and its branches, and these also contain sodium voltage-gated channels and potassium voltage-gated channels. And then we have the transmissive region of the neuron, and this is at the synaptic knobs. And here we have our voltage-gated calcium channels as well as our calcium pumps in order to help to release our neurotransmitter from the neuron itself. So if we take a look at this graphic, you'll see the neuron itself. You have the dendrites at the top, the cell body, the axon hillock, and the axon. And then you'll notice that we have these kind of color-coded areas: the receptive region, the initial region, the initiating segment, and then the conductive region of the cell. And so, same thing as before, you can kind of take a look at this and just review it for a really good or a better understanding of what the neuron itself does and what it really looks like from a functional perspective. And then here at the bottom, the last section, we get the transmissive segment. And that's again where we have our synaptic bulbs, axon terminals, and we're now able to release our calcium into the synaptic terminal to release our neurotransmitter. And this will be where we stop for today. We'll pick up the next slide as we get into our next section of lecture.